So first of all, my name is Kevin Ong. I'm a professor in the Associate Department Head overseeing extension programming in plant pathology and microbiology. So wearing that hat first, I welcome you to Texas A&M and to the plant pathology and microbiology building. You'll have an opportunity to visit a few of the labs that we have in this building. We still consider this building new, even though we moved in fall of 2019. The research wing where you'll get a chance to visit my applied research lab and Dr. Barshow's lab is in a I guess you could say a control access portion of the building. It's a USDA certified biocontainment facility. What that means is we get to play with some stuff that a little bit more interesting. But at the same time, we also have to deal with a lot more paperwork. But nonetheless, it'll give you an opportunity to see that those type of operations we have here at least relate to Rose research. Department of Plant Pathology, we are a diverse group of folks working with many different crops. Just a couple of several of us work in ornamentals, but we have folks working in all areas from cotton, corn, all the way to environmental stuff, looking at remediation and whatnot. So it's a really diverse department. So welcome to Texas A&M, welcome to this building, and let's move on. So I don't have a lot of time. When I was asked to help out Oscar to put this together, truth be told, is Jennifer and Oscar did most of the work. I just wrote on the coattails. What I get the opportunity to do is to share with you a little bit history about this project and where it came from. And this talk was actually presented last year around this time in Tulsa at the International Plant Propagation Society's Southern Region meeting. And I also want to share with you that if any of you look at this and said, hey, I want to use this in a future talk, let me know. I'll be glad to share the PowerPoint with you. So the genesis of this project, or how I got suckered into this project by Dr. Dave Burns, was the disease called Rose rosette. And I had been working some on it in the early 2000s, but I'm also a little bit of a history buff. So what I wanted to do was take a look and see what was in literature. Here's the funniest thing about disease and history. So we all read about this disease being attributed to a report back in the 1940s, basically a find in Manitoba, Canada. I actually went to look for the darn paper of the report, and all I found was a one sentence that's highlighted in yellow. And that was what it was attributed to. So somewhere, sometime in the 50s, somebody wrote about this and said, hey, that's where we got the report. And I went back to the original and it was one line. You know what? I wish I had that kind of imagination. And I hope I do. And who knows, that may have been the first, that may have not been the first, but nonetheless, it was the first recorded instance where we could say witch's broom associated with roses. Moving forward, and a lot of this reports actually came out in the 50s, in papers that are written in the 50s. So then there was an attribution that in 1941, it was found in Wyoming, and 42 in California. So all this stuff managed to find the species as well. So not your typical cultivated roses. Late 60 in California again on Rosa woodsii. If I'm not mistaken, that was up in the northern mountains or something. And at one point, Dr. Brent Pemberton had said, hey, maybe we should go, take a hike and go look for it. Thought about it, but never did it, and he retired. Let's see. Moving forward, then in the late 50s in Nebraska, at the North Platte Experiment Station, that was when there was a report on breeding stock and on large growing of Rosa multiflora. And then moving on, 76, there was a report of it in Kansas. Late 70s, you notice it goes to eastern Kansas, western Missouri. Symptoms on rose hybrids. Probably the first time that I can find that said anything about rose rosette symptoms on hybrids. And then there were also symptoms on multiflora in that report. Now, here's the fun thing. There's a paper by Allington et al., 1968 paper, where it made that statement. Just quote from it: unlikely that rose rosette will be troublesome on cultivated roses in urban and metropolitan areas. I said that I'm not ever going to be a plant pathologist who say something like that and end up, 20, 30, 40 years later, eat my words. Unfortunately, I've already made that mistake at least once with a tough grass disease. So I guess that's what plagues some of us that work with diseases. Moving forward, 82 is in Oklahoma on cultivated hybrids, and also reports from Northwest Arkansas. Now here I just kind of took a map because I wanted a map of the Mississippi River showing that in about the 80s, that's where it skipped over to the eastern side of the Mississippi River into Illinois, Kentucky, and Indiana. And what they noticed was this was report on naturalized stands of multifloras. Now, on this side of the Mississippi, in the 90s, it popped up in Texas, mainly on multiflora rootstock in the actually ornamental trade. This was in East Texas, primarily, where it first started. And my former colleague, George Philly, who has since retired, had done some work on that. And I remember he was telling me, oh, yeah, just use pylon. No, you can't use that anymore. But it pretty much killed the mites and he kind of stopped the problem. So he said, it works. That was in early 90s. Now, on the other side of the river, what we did in the 90s is it shows up in Tennessee, Ohio, Pennsylvania, West Virginia, and 96 reported in Maryland. So this just gives you a little bit of a sense about the spread of where it was first reported and how it spread throughout this country. Oh, I should also mention one thing. As far as, some people like to say, so where did this disease come from? This is inherently, as far as we can tell, a North American disease. I had not seen any credible reports of rose rosette from any other continents yet. I have suspicions of where they are, but none of it credible. Yeah. There have been at least, I know, 2 reports that I've seen from India and 1 report from Russia. But again, that's not enough information that would suggest that it is in fact rose rosette disease the way that we know it here in North America. Now, a little bit of the science spark, real quick to go through, is in the 80s is when they start getting some evidence to demonstrate that it is some form of biological agent, and it works and acts very much like a virus. And in 2011 was when this virus was characterized by Alma Lady, who was a master student in Yanis Tazanakis' lab at that time. It was kind of interesting. I was working with rose rosette in the early 2000s. And the way that we would diagnose it is if you see symptoms and you find the mite, it's a positive. It wasn't until after 2011 that we actually had a genetic tool to be able to go after that virus. And one of the things about having the ability to go that, we also learned that that's not always foolproof. And what I meant by that was, I remember when we had that first tool and we were using it, ever so often we would run into a batch of plants that the test wouldn't work. And so off we go to find yet another universal thing and realize that there were some nuances in how we are able to detect this virus. To make matters interesting, and I get a lot of questions on the extension side is, gee, who induced this spread? And so Dr. Jim Embrine, who is Dr. Mark Wyndham would call him a mythologist, is a carologist, a mite person, basically said, or at least quoted him on this, virtually certain that rose rosette will greatly reduce the density of multiflora rose. And that was in the context of utilizing rose rosette as a biological agent to manage multiflora rose, which is considered a nuisance plant. So it was utilized by the federal government in some areas as a plant control agent. And in one sense, it is an explanation of how this thing got spread around the United States in a pretty rapid fashion. Moving forward, 2014 was when the first SCRI project called Combating Rose Rosette Disease came about. And 2023 was this project that we're currently in, developing sustainable rose landscapes. Now, real quickly go through to make sure that everybody's on the same page about rose rosette and the how and who and what caused it. Current knowledge, the vector of the virus is an aerophyte mite, a microscopic mite, Phyllocoptus fructiferus. As far as we know, the virus is restricted to the genus Rosa, where it does damage on. As far as we know, all rose varieties are susceptible. Doesn't mean, in other words, they're not immune. Are there resistance? Very likely there are some levels. The part about typically kills roses, that's always been an argument between me and Dr. Mark Wyndham. Dr. Wyndham usually says in Tennessee, it will kill it within that year or 2 years in the winter. And I keep telling him, I said, I've seen it in Texas where I can see it, living for 6, 7, 8 years. And part of it has to do, I think, a lot with the ability of the plant to be able to keep carbohydrates and surviving through some of the harsher winters. So bear in mind the 3 components. 1, the virus, rose rosette virus. 2, the vector, the aerophyte mites. And of course, you need a host. Now, to throw in a wrench in this, in 2023, Tobias de Sierra had a paper published and reported on a second mite vector, Phyllocoptus arcani. Depends on who you talk to. Some says that's a true species. Others said it is a sport of Phyllocoptus fructiferus. So take your pick. So as far as general knowledge right now in literature, what we do know is transmission of this virus is by Phyllocoptus fructiferus. So are there other potential modes of transmission? Very likely, but none that is published or shown at least scientific data on. Out here too, I want to ask, I guess, some of our show students, hey, if you guys got a new image of the virus for the rose rosette virus, I'd love to get it from you guys. Just to give you a sampling of some of the symptoms here. Rose rosette is not one of those easy ones to deal with out there. I think the extension group realizes that you could have one, you could have none of the symptoms, and you could still have a positive plant. And the symptomology is quite variable, depending on not only species, but I think it's also regionally involved. Now, real quick to kind of highlight, then I need to move on, is to show you a little bit, a compare and contrast the two projects. So the four objectives of that first project in 2014, the Combating Roads to Rosette, is listed above. But I just want to draw your eyes to those two things that I highlight, because what I highlighted was what I said we somewhat achieved at the end of that project. The ones that are not highlighted was what we would have liked to have gotten, but I think we didn't quite get there. So what came up was this new project here, was what Oscar went over with you guys, and you might realize that some of this really addressed, like number 2, additional resource of resistance. That's still part of number 2. One is we're still trying to figure out, learn a little bit more about the virus itself. My efforts and all that is primarily focused on developing a comprehensive research, demonstration, and education program. And what that is, short term. We want to look long term, but in the short term, people need some answers. People need some solutions. What do we have right now? What's the best that we have? What can we show? What can we tell folks? And so that's what the extension group does. So moving forward, I'm going to skip some of this. So this was some of the questions that I shared with that group last year. Like, where did this disease come from? Well, what will the rest of the roses get infected if it's in a garden or whatnot? What's the correct action for management? We don't have a great answer for that. We're trying to. And could this be prevented? So those were things for them to ponder, and perhaps for you to ponder as well. Now, I want to quickly go through and talk a little bit about the current cycle and the stuff that we're doing as an extension group. And I'm thankful that some of you are here and to share what we have been doing the last couple of years or so. So when we put this thing together, we said, we wanted to do research demonstration. So in a way that we can put things out there, let people see it, let them draw their own conclusions from it, but we have the opportunity to influence what they think, what they see, because we demonstrate stuff, very tentative extension. Beyond that, we want something out there that we have the ability to capture some data and make sense of it in a scientific fashion. So it's just not anecdotal, but we are hoping to get some hard data. So how do we go about putting something like this together? There were actually 3 different modules that we put together. I'm just going to share with you about the resistant cultivar trials, and Dr. Runabout will talk to you a little bit about the miticide trials, the modules that we are also doing. So real quick to run through the black spot and rosette resistant trial. It's a trial, and this was basically excerpts from the modules that we tried to share with county agents, with other folks, that said when we tried to solicit for cooperators. We're looking at 3 to 5 plants, each of 5 to 10 cultivars in the field. That's what you put out in the field with resistant to rosette disease or to black spot disease. And here's the kicker. We have to always tell folks, we always have to put a control. And control means we know that something is going to get hurt, because if we don't know it gets hurt, then we don't know what's going on. And that's always the challenge when working with the general public. In terms of experimental design, nothing less than 3 replications. Why? Statistics. We need that. Management of roses. Here's the thing. We realize with roses in the general public, different folks choose to care differently depending on where they are. And so in terms of when we look at this, we said, you know what? Some of these folks, whether they're commercial landscapers or parks and rec municipal type landscapers, they have a way to manage their land and their plants, because whether it's budget or whatnot, and we want to see how these plants do in nature, quote unquote, natural type situations. So we said, However you do it, it's fine. We just like to have access to that information, so we know if you did use fertilizers, what kind and when, if you did use pesticides, what kind and when, if you did prunings or whatever, when. And so just to at least have some data, and we take that as potentially negligible actions to the data that we would want. Kind of show you that the list that we initially put together, thanks to Dr. Dave Burns helping this out, we had about 20 or so, and we said, this are the 20 that we're going to look for. Well, guess what? When we started looking at the cultivars, those cross out we never did use. Those that we did, this is sort of a list of the cultivars that we did use. Now, not every one of this cultivars at every location where we have our extension researchers and cooperators are. So it's a subset. And I'm not going to share with you, but the way that we collect data, not only where we put our grad students or ourselves pulling data on an Excel sheet, but we actually do it electronically online through an app. And hopefully Joe LaForest at University of Georgia will get a little chance of explaining that. But we also have a module that we try to engage the general public in terms of capturing some citizen science ability, and it asks them 4 questions: 2 on how you rate rose rosette, and 2 on how you rate black spot. Why only 4? Well, we need that. I figured once you get more than 4 questions, you lose them. They lose interest. So you want to capture them real quick, and it can be done at any time. So with that, I'm going to quit right here because I'm right up at time.